Dear students, please note down. The notes of this video lecture are available in the description of video. Please download your notes. Dear students, in today's lecture we will discuss urea cycle, ornithine cycle, Krebs and Hanseleit cycle. That is an important topic of chapter excretion. Note down, the students of BSc second year 4th semester students, final exam topic, that is urea cycle, ornithine cycle or Krebs Hanseleit cycle. As we know, in our body the proteins undergo oxidation, and oxidation of proteins in our body produce amino acids. These amino acids undergo deamination. As a result, some toxic compounds are formed like ammonia. In our body, ammonia is highly toxic, and this toxic compound is produced in all body cells, especially in liver cells. Note down, dear students, all competition students, in liver cells, the mitochondria of hepatocytes that converts this highly toxic compound ammonia into less toxic form, that is urea, and this entire cycle is named as urea cycle, that was described by Krebs and Hanseleit in 1932. So this process is named Krebs and Hanseleit cycle. This entire process that is divided into 4 steps. It means the mechanism of urea cycle involves 5 steps. First step is formation of carbamyl phosphate. Now down, all competition students, in this first step, carbon dioxide, this CO2 interacts with one molecule of ammonia, and in presence of this enzyme carbamyl phosphate synthetase, and a cofactor which is needed in this step, that is Mg positive 2 ions. This step also requires 2 molecules of ATP, and this CO2 in presence of this ammonia that produce carbamyl phosphate, which is also named carbamyl. Second step of this mechanism, urea cycle, that is formation of citrulline. Formation of citrulline. This carbamyl phosphate interacts with ornithine molecule, and in presence of this enzyme ornithine carbamyl transferase, this carbamyl phosphate is converted into citrulline. This step produces one molecule of water. Dehydration occurs. Third step is formation of arginosuccinic acid. First step is formation of carbamyl phosphate that involves interaction of one CO2 with ammonia. In presence of this enzyme carbamyl phosphate synthetase and cofactor, that is Mg positive 2 ions. This step also requires 2 molecules of ATP, and it produces carbamyl phosphate. In second step, that is formation of citrulline, in which this carbamyl phosphate interacts with ornithine molecule, and this step is catalyzed by ornithine carbamyl transferase enzyme. It produces 1 molecule of water and 1 molecule of citrulline. In third step, that is formation of arginosuccinic acid. In this step, this citrulline combines with aspartic acid in presence of 1 molecule of ammonia, 1 molecule of ATP, and this step is catalyzed by an enzyme, that is arginosuccinate synthetase, and the cofactor which is needed here, that is Mg positive 2 ions. It produces 1 molecule of water and 1 molecule of arginosuccinic acid. Dear students, the fourth step that is formation of arginine. This arginosuccinic acid, it is converted into one molecule of fumaric acid and one molecule of arginine, and this step is catalyzed by an enzyme that is arginosuccinase. This fumaric acid is converted into oxalosuccinic acid, which is transaminated into aspartic acid, and this aspartic acid combines with this citrulline to produce arginosuccinic acid, and this arginine that gives rise one molecule of ornithine. This is last and final step of urea cycle, formation of ornithine. This arginine in presence of water molecule, and the step is catalyzed by an enzyme that is arginase, and this arginine in presence of this enzyme arginase and one molecule of water, it gives rise one molecule of urea and one molecule of ornithine. This ornithine repeats the cycle. It means in this entire process, one molecule of CO2 and two molecules of ammonia, one molecule of ammonia in step number first and another molecule of ammonia step number third. It means this entire cycle involves, all competition students note down, two molecules of ammonia and one molecule of CO2 to produce one molecule of urea. This entire process takes place in liver cell hepatocytes, and this cycle is carried by mitochondria of liver cells. Dear students, this is urea cycle. In this ornithine repeats the cycle, so it is named ornithine cycle, and this entire process was explained by Krebs and Hanseleit, so it is named Krebs and Hanseleit cycle. Dear students, this entire process that takes place in liver cells, hepatocytes, which is carried by, note down, by mitochondria of liver cells. In this process, the most toxic compound, that nitrogenous compound, basic catabolic product of amino acids, that is ammonia, and this most toxic compound ammonia is converted into less toxic form, that is urea, and this process takes place in hepatocyte of liver cells, which is carried by mitochondria. This entire process involves 5 steps. First is carbamyl phosphate or formation of carbamyl. This step involves one molecule of CO2 and one molecule of ammonia in presence of 2 ATP molecules, and the reaction is catalyzed by carbamyl phosphate synthetase that requires a cofactor which is Mg positive 2 ions. Step number 2, that is formation of citrulline. In this step, this carbamyl phosphate interacts with this ornithine molecule in presence of ornithine carbamyl transferase enzyme, and this enzyme catalyzes this carbamyl phosphate and ornithine into citrulline that also gives one molecule of water, and this citrulline interacts with aspartic acid in presence of ATP and one molecule of ammonia, and this step involves an enzyme that is arginosuccinate synthetase and the cofactor that is Mg positive 2 ion to give rise arginosuccinic acid. This arginosuccinic acid, it is converted into 2 molecules, one is fumaric acid and second is arginine. This fumaric acid is transformed into oxalosuccinate and it gives rise aspartic acid. And this arginine, in presence of this arginase enzyme and water molecule, to give rise one molecule of urea and one ornithine. This ornithine repeats the process, and this product urea we can retain, we can store this less toxic form, that is urea. And this entire process, that is urea cycle, which takes place in liver cells, hepatocytes, which is carried by mitochondria of liver cells. Dear students, this most toxic compound ammonia, we cannot store it, so it is converted into less toxic form. If this process fails to produce urea, then this toxic compound, it damages our body cell tissues, particularly our brain, and it becomes fatal. So it is converted into less toxic form urea. We can retain this urea for some time, and we remove urea as a chief excretory product. So we are ureotelic. This is all about urea cycle, ornithine cycle, that was described by Krebs and Hanseleit. So it is named Krebs and Hanseleit cycle.